Well, good morning, Biology 20s. So today we are going to discuss enzymes. We've talked about digestion and how enzymes are important, but what are enzymes? Well, today we're going to learn about some important keywords, and our concepts here are going to be all about enzyme activity. What is an enzyme? How do they work? What are different factors that would influence their activity? That's going to be our focus today. So first of all, enzymes are proteins. That's the class of macromolecule they belong to. And you guys know already that amino acids make proteins, and proteins are really huge molecules, right? Hundreds, thousands of atoms, and they fold and they make these really complex molecules. And so enzymes are proteins. Okay, so typically when we draw a diagram of an enzyme, we draw them like a little Pac-Man here. And so here's our enzyme. I'm labeling enzymes E. And then the substrate is the thing that the enzyme acts on. Now, this is pretty specific, right? Every enzyme has its own specific substrate. So for instance, if its job is to break down sugar, sucrose, disaccharide, it's not going to act on proteins. It's not going to act on fats. It's specific to that particular substrate. So when the enzyme and the substrate get together, the active site is where the substrate fits in. So I highlighted that there in yellow. And so they fit together, and you'll read that it's kind of like a lock and a key. So we've got pictures here of what an actual enzyme looks like. Like these are the molecules that you can see, carbon and hydrogen atoms, all of these little round dots here. You can see how massive the molecule is. And the substrate, again, that's the thing the enzyme acts on. The active site we showed as Pac-Man's mouth, and that's where the substrate fits in. Okay, so again, how does an enzyme work? Well, it's just like a lock and a key. And so as we just said, the enzyme is able to fit in the substrate. So it might be two substrates coming together to form one compound. So there's the substrates, and they're joining on to the active site. And just like key, so here's the key, the substrate, here's the lock, the enzyme, they fit in perfectly. And then we have the enzyme substrate complex, and it is able to do its job. And what's its job? Well, it is going to make a product of some sort. It catalyzes a reaction, and then the product is created. It can go off into the cell and do whatever it's supposed to do. And then here's the enzyme. It's able to go ahead and catalyze that reaction again. And so it can repeat the reaction over and over again. It has not been consumed by the reaction. It is able to keep on working. So scientists have changed this model a little bit. They've modified this, and now actually they talk about the induced fit model. And here's what's different. When you still got your substrate binding to the active site of the enzyme, but now the enzyme is undergoing a shape or a conformational change. And it's kind of, I think of it like it's an enzyme hug. The enzyme is going to basically just fit a little bit better with that substrate, and then it catalyzes the reaction. Here the products are made, and the enzyme can catalyze these reactions again and again. So I've used that word a lot, catalyze. It comes from the word catalyst. What is a catalyst? Well, a catalyst is a molecule that speeds up chemical reactions. It's going to make the reactions occur more quickly because your body reactions are taking place at a pretty low temperature, right? Like 37 degrees Celsius is a lot lower than some reactions are occurring in industrial processes and stuff like that. And so these reactions need to occur with an enzyme catalyst in order to speed up the reactions. So there's two major classes or types of reactions, and those are catabolic versus anabolic. Either you're breaking apart molecules, you take a large molecule, a large substrate, and you break it down into two smaller products. And examples of that is like in digestion and cell respiration, and typically energy is released exothermic or exergonic. Anabolic, on the other hand, is where you have two small substrates and they come together, and then the enzyme catalyzes and it makes a larger product. Now, the way I remember this is anabolic steroids. Oops. Sorry about that, my alarm going off. I remember anabolic steroids, and bodybuilders would use these to build up larger muscles. They're totally illegal, right? They're not something that bodybuilders or anyone is supposed to take. They're performance enhancing drugs. But how I remember it, anabolic is building up. And so you have to absorb energy to make larger molecules. Okay, so again, enzymes are catalysts. And so without an enzyme, the amount of energy that you need to kickstart a reaction is a really high amount of energy, right? So without enzymes, you need a lot of energy. But then when you have an enzyme, it reduces that activation energy, and now it catalyzes the reaction. It stimulates the reaction to occur at a much lower energy level than if you didn't have the enzyme present. Okay, so moving on. One more thing I wanted to talk about is enzyme. How to recognize what an enzyme is, its name. And so typically there's an ending ASE, the suffix. So carbohydrates breaks down carbs, amylase, lipase. And that's not always true. You guys know that like pepsin is an enzyme found in the stomach, breaks down protein, and it doesn't have an ASE ending, but a lot of enzymes do have that ASE ending. And so recognize that if you see that, that's typically going to be an enzyme. All right, factors affecting enzyme activity. So looking at this graph, we have to understand that enzymes, because they're protein, they're pretty sensitive to their environment. And so what happens in their environment can affect the rate of reaction, the activity, how active or how able to catalyze a reaction the enzyme is. So you'll notice this graph in temperature, it's kind of three different parts to the graph. And as we see, as the temperature is increasing, there's more collisions between the enzyme and the substrate. And the reason why is the molecules have a higher amount of kinetic energy, right? They're moving around, they're colliding with one another, the substrate and the enzyme bounce into one another. And so as the temperature goes up and the activity of the enzymes goes up, well, the reason why is because there's more collisions. They bump into one another. And then you can see it reaches a maximum around 37 degrees Celsius. That's body temperature. And that's the optimum, the best rate, the maximum rate. That is when enzymes do their best work in our body. And then you would think that, well, if we keep increasing the temperature, we got all this high kinetic energy that the molecules, the enzyme and the substrate would collide more. Well, actually, that's not true. The protein actually begins to denature. Remember, again, enzymes are protein. And so now what happens is the active site starts to unravel and unfold. And when that happens, the substrate is not able to fit anymore. And so the activity actually decreases. Now, fortunately, that's reversible, but it is going to change the shape of the active site. Okay, so next we talk about pH. And pH is how acidic or how basic, alkaline the surroundings is for the particular enzyme. So when we look at our pH scale here, anything below 7 is an acid and above 7 is a base or alkaline. So you can see again that enzyme activity increases, enzymes and substrates have collisions, and they like a certain pH, and then they reach a maximum, and that's again their optimum. And then as the pH changes, it's a very narrow range for pH, the activity decreases, and then that enzyme again is going to denature. Okay, and so you can see the optimal pH for a lot of different enzymes. So like this guy here is around 7. This guy, well, obviously he's in the stomach because that's such a low pH. And most enzymes in your body are going to be around pH of 7, but there are some that like a higher or a lower pH. And so you can see what their optimum temperature is, too high or too low of pH, and that enzyme will start to denature. Okay, so next up is substrate concentration. And so as we increase the amount of substrate, Here we see kind of two parts on the graph. Number one is as the substrate, what the enzyme is working on, concentration goes up, the rate of reaction, the activity of the enzyme goes up. Again, why? More enzyme substrate collisions. But then you see in the second part of the graph here, section 2, that the graph kind of flattens out. It plateaus. So the maximum rate, it can't kind of get any higher than that, because at that point of time, the enzyme becomes the limiting factor. And there's so much substrate, but not enough enzyme. And so you can see here's all these substrate molecules, but when an enzyme is busy catalyzing, well, here he is, and he is not able to catalyze any more than one reaction at a time. And so enzyme has now become my limiting factor, and that explains why the graph plateaus. So enzyme concentration is going to be a direct relationship again, but again, if there's not enough this time substrate present, so here's my increase, just like this graph is showing, more and more enzyme substrate collisions, but this is if there is as much substrate present as we will allow. So if we keep adding substrate, then the reaction keeps increasing. But if we have not enough substrate, we've got too much enzyme. So that's what we're seeing over here. We've got lots of enzyme, not enough substrate, and our level is going to plateau again. Okay, coming back for one more video on inhibitors and talking about the types of inhibition. So that'll be just a short little video, and we'll see you again in just a minute. Take care.